really getting into Pingu. But anyway, so Emily, last time you and Shelty were in, she was having some eye problems, wasn't she?
Yeah, that's right. Do you have any good news for us?
Well, we've got the reports back from the ophthalmologist, and it's not quite what we were hoping for. It turns out that Shelty is suffering from a disease called progressive retinal atrophy, or PRA, and that explains why she's losing her eyesight.
Okay. Is that as bad as it sounds? Is your eyesight going to get any worse?
Well, unfortunately, yes. PRA is an umbrella term for a group of retinal diseases that have a gradual onset, so it will get worse with time. Now, how much do you know about eye function?
I went to an ophthalmologist once, but that's about it.
Well, the retina is the light sensing region of the eye, right? So when you get retinal atrophy, there's loss of function in the cells in the retina, and this ultimately causes the retina to stop working properly, so you lose vision.
Okay. So how did my little Shelty get this disease then?
That's a good question, actually. I remember learning about this back in vet school many years ago.
Excellent. So you should all know that PRA is a fairly broad term that encompasses a variety of mutations throughout various breeds. So this particular case involves a 5-year-old female spayed Sheltie, or Shetland sheepdog, and she's been showing signs of progressive loss of function of her rod photoreceptors leading to nighttime blindness. Now, this 2015 study published by Animal Genetics identified a novel mutation in a particular gene that was responsible for PRA in some of the Shelties. Does anyone know what this mutation was? Samuel Leo?
I think the authors decided that the CNGA1 gene was a likely candidate because it's in the manus pigmentosa in humans, which is analogous to PRA.
Yeah, that's excellent. So yeah, CNGA1 encodes for the cyclic nucleotide gated channel alpha 1 in the rod photoreceptor cells in your retina. So the resulting transmembrane protein is basically a cation channel that allows for depolarization of the rod cell in response to light in a process called phototransduction. So that's great, but do you know exactly what the mutation was that they found?
It was a 4 base pair deletion. Yeah. So it was a nonsense mutation because there was a frame shift in the gene, making a premature stop codon. So the transporter protein becomes shortened and it doesn't work anymore.
Yeah, so that's exactly it. So in Shelties they observed the CNGA1 mutation. However, there's another mutation in dogs that causes PRA that's more common, and this is known as PRCD, which aptly stands for progressive broad cone degeneration. So this mutation in the gene again causes the associated protein to become shortened and non-functional, except we don't really know what the original function of that protein was. Either way, you see a collapse of nuclear and photoreceptor layers, loss of ganglion cell layer, and complete loss of photoreceptor nuclei. So let me ask you, with the Sheltie with PRA, how could you tell if it was due to a mutation in the CAG1 gene or the PRCD gene?
Maybe some sort of genetic test.
Yeah.
Yeah.
Pretty good. It's pretty good. So we can take a blood sample to test for the PRCD mutation and obtain a genotype for that particular gene. However, there's no test for the CNGA1 gene mutation. If the dog does have PRA, you can be fairly sure that it's either homozygous for the mutated CNGA1 gene or the PRCD gene. So why might that be?
Because they're both autosomal recessive.
Yeah.
Yeah, so the dog has to have both defective alleles for the disease state to be apparent.
Yeah, that's exactly it. So, do you know about any treatments for PRA?
This was in the lecture a few weeks ago. Something about viral therapy.
Just a few more minutes and then we'll have a quick break. So I'll briefly talk about some of the examples of viral vectors used to treat diseases. A study that looked at the treatment of retinal diseases determined that recombinant adeno-associated viruses were the most suitable for treatment of retinal diseases. A 2001 study that used RPE65 deficient dry eye dogs, which consequently suffered from retinal dystrophy, found that after subretinal injection of the recombinant adenoviruses containing the RPE65 gene, their retinal function and electroretinography results improved.
Yeah. I remember. So you can use empty viruses for gene therapy and use the viruses as vectors to deliver the DNA or RNA, and it encodes a normal or corrected protein. So this amends or protects against the genetic disorder.
Yeah, that's correct. So remember that viral delivery is a relatively uncontrolled process and is associated with some significant safety risks, including adverse immune reactions, mutagenesis, or reversion of the virus to the virulent state. And any beneficial effects are usually not long-lived, particularly with adenoviruses or adeno-associated viruses, as they introduce RNA or DNA, is not integrated into the host cell's chromosome.
So viral gene therapy isn't a good option.
No, not with current methods. It's generally considered that the risks of gene therapy outweigh the potential benefits. Unfortunately for us and our Shetland sheepdog, there is another option though. And this is encapsulated cell therapy, or ECT. So this involves cells that are genetically engineered to produce and secrete a therapeutic factor. In comparison to viral therapy, it's a more controlled and persistent treatment option, and the encapsulated cells can be removed at any time.
Damn, Miss Frankel, that sounds crazy.
Yeah, it's pretty incredible. A study conducted in 2002 looked at prolonged intraocular delivery of the ciliary neurotrophic factor, or CNF, in the treatment of a particular form of PRA in dogs through use of ECT. They found that the eyes that received the treatment retained a greater number of photoreceptor nuclei in the outer layer of the retina in comparison to the untreated eye, which degenerated as expected with PRA.
Okay. I think I get it all. I just got one more question actually.
Yeah.
Is any of this going to be on the exam? Oh. Yeah, I remember.
So to start off with, progressive retinal atrophy is a genetic condition. So it's incorporated into your Shelties DNA. So I guess first I'll explain what has happened to your Shelties DNA and how it's causing the vision loss. And then I guess we can get into some possible treatments for Sheltie. How's that sound?
Yeah, that sounds great.
Alright, so as I mentioned, it's a genetic disorder that affects the eyes. So your DNA is basically like an instruction manual, and particular genes tell your cells how to build the structures within your body. So when a mutation occurs that significantly changes these instructions, you can get proteins made that are non-functional. So what we think has happened in Sheltie is that there's a mutation in a gene called CNGA1, or it might be a gene called PRCD, we're not sure. But what has happened is that there is a non-functional protein being produced. So without this protein, visual information that reaches the retina can't be converted into an electrical message, and therefore it's not delivered to the brain. So sight is lost. Are you following that so far?
Yeah, I think so. So is there anything we could do for Sheltie? Or like, how long until she loses all again?
It's actually very difficult to determine how long it will take for Sheltie to lose her vision, since it's a progressive disease. The rate of progression can change between individuals or between breeds, so it's hard to give a definitive answer. However, for treatments, there isn't much that can be done to restore any vision lost already. But we do have a few strategies you can put in place to make sure Sheltie has the best quality of life possible from here on out.
So there's nothing that can be done to restore the vision?
Well, there is some research into new treatments that involves inserting special cells in the eye, and these produce therapeutic factors, and they supposedly preserve the remaining functional cells. However, it doesn't restore any vision lost already. There's also some research being conducted into inserting the correct functioning genes via viruses into Sheltie's cells so that the protein is produced correctly. However, all of this research is just experimental at the moment and probably won't be available for treatment for quite some years.
Well, yeah, that's fair enough, I guess. A couple of years ago, Shelty had a few pups with a cattle dog. I was just wondering if they'd be affected.
That's an interesting question, actually. Maybe you should come over here and have a look at this Punnett square I keep drawing up. I've spent your time.
Okay, so you've done biology, right?
Yeah,
you
yeah.
know about, recessive alleles and dominant alleles.
Yep.
So basically, this is a recessive disease. So you're going to have two recessive alleles if you're in the disease state, heterozygous for a carrier, or two dominant alleles for normal. So with Shelty, she's diseased, so we know that she's got two recessive alleles. Thing we don't know is the cattle dog. We don't know what genotype it has. But let's say, just for example, it's got a dominant and a recessive allele. Then the offspring would have 50% carriers and 50% diseased. But the thing that makes this more confusing is that we don't know if Shelty has the CNGA1 mutation or the PRCD mutation. So if it had the PRCD mutation, then what offspring you would get, whether carriers or disease, would depend on the genotype of the cattle dog. But if it had the CNGA1 mutation, they would all be carriers. Now, do you want to come back to the desk? So, all of these pups, you could determine their, genotype through genetic testing, but the thing is, there's only a test for the PRCD mutation. There's no test for the CNGA1 mutation. So, I think the best option with these puppies is to monitor them closely, give them regular eye tests, just get onto any loss of vision early so you can act accordingly. I just would advise against breeding with these pups in case they're carriers for disease.
Just one last thing. With Shelty, is there any management advice you have?
I think with Shelty, from now on we'll just try and make her life as easy as possible. So if you just stay in the same house, that's best, or just try not to move furniture around too much, just so she's familiar with the layout and where everything is, so she's not running into things. You can also get these collars which have little bumpers on them so she can sense when she's going near things, so she doesn't run into them and hurt herself. That's what I would suggest. So do you have any other questions? That all makes sense?
No, I think that's about it, thanks.
Oh, actually, I remember we had this brochure too on how to look after blind dogs we give out. Do you want to take that down maybe? Have a
That's
read?
great. Thank you.
Oh, yeah, awesome. So that's it.
Yeah, that's
Oh,
it.
thank you. If you have any other questions or concerns, just ring me up anytime. That should be all right.
We'll do. Thanks for that.
Have
See
a good
you.
one.